Welcome to FlowStars, candid conversations between Dr. Peter O'Toole and the big hitters of flow cytometry, brought to you by Beckman Coulter at Bitesize Bio.
Today on FlowStars, I'm joined by a special set of guests to talk about spectral flow cytometry. We're going to talk about all the advantages that can enable us as flow cytometrists and as science going forward. Hi, I'm Peter O'Toole from the University of York, and welcome to FlowStars, a special edition today because we're now focusing not necessarily on the people, although we will talk to the people, but also we're focusing on spectral flow cytometry and the new instrument from Beckman Coulter, the Mosaic, or the add-on. We'll talk about that as we get there. But first, let our guests introduce themselves. So Domenico, please, will you kick off?
Yeah. Hi, everyone. I'm Domenico Tartaro, a postdoc immunologist seated at Modena University. We already have the Cytoflex LX. Now we have upgraded with Mosaic, and we are focused on the T-cell immunophenotyping. So we have this kind of expertise.
Domenico, thank you. Andrew.
You're welcome.
Hello, everybody. I'm Andrew Patentreger from the Broad Institute in Cambridge, Massachusetts. I've been here for 3 or 4 years now, and we have plenty of Cytoflexes at our disposal. So this has been a great addition to the lab. And while I don't think I'm quite an expert, I feel as if I have a good bit of experience in flow cytometry while managing this flow core.
Andrew, thank you. And John?
Hi, everyone. I'm John Bianchi Colangeli. I'm at the Broad Institute Flow Cytometry Facility. I'm a research associate that works on immune profiling studies where we do a lot of spectral flow cytometry and single-cell RNA-seq. So I've been here for 2 years doing lots of spectral flow and working with Andrew and learning as much as I can.
John, thank you. Tamar.
Hi everyone, I'm Tamar. I'm an immunologist by training and a bit of a flow nerd throughout my whole career, which led to me being now the head of the flow cytometry core facility at the Leiden University Medical Center in the Netherlands. So I'm the head of a large flow core that has a lot of spectral instruments. All that knowledge we're trying to use to put the Mosaic to its, to test it to the limits.
Tamar, thank you. And Kelly.
I'm Kelly Lundsten. If Tamar is a flow nerd, then I'm a fluorophore nerd. Usually that's my contribution. I am a consultant for Beckman Coulter on the Mosaic project, and I work for Fluorofinder part-time as well.
So you're the colourful one out of us then?
I like that.
I liked earlier, actually, in our discussions before we all started, that Andrew mentioned that he's got a multifunctional unit. Was it, Andrew, you mentioned?
Yeah, absolutely. So while we house flow cytometry instruments, we also have genomics instruments such as sequencers. We also have a tissue processing core with microscopy and confocal microscopes. So we do quite a bit. I manage the flow cytometry portion. But we're all kind of intermingled throughout each department.
So very similar to York, except you were very kind to call yours multifunctional. I call mine dysfunctional. I'm joking, not really. So actually, this is really timely because I'm also very fortunate. So my Cytoflex Mosaic has arrived at York. It's set up, it's running, but we haven't had any time to get on it. It literally has just arrived. So I am going to learn so much today about what else we can do with it and what your thoughts are, what the advantages are, and everything else around it. But maybe I can start off with why spectral? Actually, I'm going to go to fluorophore geek first. Why spectral? Kelly?
Yeah, I'll take that one. I was just writing a blog on voltage setting and how we used to do it. And spectral is such an important advancement. It doesn't matter whether or not you want to do 30 things. It's still important that we're assessing the full spectrum and we're seeing, everything that we can see. So obviously the biggest attractive feature of spectral is getting up to 30 parameters without as much of a fuss, without as much trouble. I remember the first time I ran my very first Aurora assay, and it was just like amazing. I could do 24 colors without a tremendous amount of effort. And I see that with the mosaic too. I mean, usually you go through all these rounds of optimization. It isn't that isn't still absolutely necessary. We're not, the first assay out of the gate is not going to be the right one necessarily. It just doesn't work that way. But how quickly that we're able to apply the new reagents to better clones, and just the process and the long work that we've all done towards optimization is definitely making it easier for just to facilitate these high parameter instruments and make the process a lot easier for ourselves and our end users. So definitely parameter level, but for me, it's also that every time when we were only monitoring the peak, we really weren't getting all the information, that we could have. And we were also implementing a lot of bad practices that I think are creating a lot of discord in our data that we were ignoring a lot. And now you can't hide bad practice from Spectral. So it's going to hold us all to a standard.
So do you think the quality of data is therefore better?
No, absolutely not. The more parameters that you implement, I would say that one of the things that's going to be really important in the future of Spectral is that we start to recognize that it's not a magic black box, and that we have an expectation that data should still perform and have a certain resolution. But that's for us to implement, right? As users, that's the manufacturers are doing their best to give us these tools.
So, Tamar, to you've got 500 users, was it? I think you said there.
Yes.
So what do you see as the advantages for it?
I absolutely agree with Kelly that it's a lot easier to design a panel for a spectral instrument. So when I was a postdoc, we got our first spectral instrument, and I've never gone back to the conventional instruments that were at the facility, because it was just so much easier, no voltage settings, it would work out of the box. A lot of our users, so they have very precious material. So that would be biopsies or some they would only have like maybe 10,000 or 100,000 cells in one sample. So by being able to go to that many parameters, you can just get so much more information out there. And most of the PIs that I know, you tell them, I can make you a panel with 40 colors, and they say, great, here's a list of 70 markers, put them in there. And but yeah, but that's how it goes. And well, at least now you can put 40 of them in your panel.
How many users, can I ask, how many users are now actually pushing up to 30, 40? It's not very, but how many are?
Oh, I think in our core, there's at least 50 people that go above 30 colors. Maybe, so we have 250 Spectral users. I think at least 50 go over 30. And 20 to 25 that go to 40 or higher. And I hear a lot of people say, well, my panel's only 30 colors, but I still think it's very interesting. And they're completely forgetting that 30 colors was impossible a few years ago.
Yeah, and the cost saving, time saving, sample saving, so all those advantages wrapped around it. So, Domenico, if I come to you, are there any other advantages that you've noticed with it?
So, I tested some stuff during the last weeks. So I also saw that there is a great improvement in signal to noise using Spectral. So I tested 2 panels of ours, and for a lot of markers, we saw a lot of improvement on signal to noise. So this can help us to find also the dimmest population, and that probably in the past years I missed during my acquisition. So probably this is one of the most important stuff that I saw now. We have planned to test 2 panels now in our lab. One is 21 colors to analyze the phenotype of teals, and the other one is designed to analyze all the T helper and cytotoxic T cells in the periphery. and the second one is 29 colors. So, but it is specific. Both of these panels are specific for T cells. So, so it's very difficult to push over 30 color this kind of market. So it's very easy to reach 40, 50 parameters when you analyze B, T, monocyte, DC, and all the population that you have in the blood. But it's very, very, very difficult to push more than 30 color when you are analyzing specifically T cells. And we are doing that. So, and in specifically, I will work and with the help of the Kelly and Beckman team, I would like to design and a new panel to analyze the immunometabolism, but this is very, very difficult step. So we can talk later about that because it's very difficult. So I'm still fighting because I'm testing the antibodies. So, but I need to understand how can I also find this kind of markers already conjugated, or I will conjugate by myself this kind of antibody.
Okay, so Kelly first, and then I'll come to Andrew and John. Kelly.
There's like a point that Domenico made that I think is really important, which is the reagents have to match where the instrument is, and the algorithms for unmixing have to match all of it. It's not like we're just waiting on instruments to get better. We're waiting on mixing to get more accurate, so our spreading error is more reduced. Our data quality is definitely being impacted by more parameters. But that can be helped by the next order and the next generation of floors and the next generation of more accurate unmixing, so that those correction factors are, contributing. So, I mean, it's a whole community thing, right? To make data that's worthy of having at a plus 30 parameter level, and also an expectation that we have of each other, that when we see data that clearly has unmixing errors, that we step up and say, that's not supposed to be that way, So I think a lot of things are coming together to make it more reliable, but it's certainly not at a place of perfection.
Yeah.
But I don't think the wellness compensation was a wellness of perfection either. So always worth just remembering that. And especially when you get to more than 3, 4, 5 colors, it starts to layer up the complexity. Or it's not really complex, but you start to see problems creep into the data. Andrew, John, I don't know who to ask, because you're sort of working, I presume you're working quite closely together in the core.
Yeah, so I'll just say something real quick, then John, you can really get into the depth of the topic. John has a lot more experience in Spectral than I do.
Only a shallow bit of the depth.
Okay, yeah. It's quite ironic. I've trained people on Spectral instruments, yet this is the first time I've actually had the opportunity to sit down and run one myself. So we ran some small panels that Kelly had designed for us, and the difference was really night and day between the conventional mode on the instrument and the Spectral mode. So that's my 2 cents on it. John, I'll let you actually get in, a little more detail there.
So you are seeing a difference in resolution?
Yeah, absolutely.
John?
Yeah, so when I started 2 years ago, I am kind of like an aspiring, or like, I guess, blossoming flow nerd. I really love it. But when I started, I actually learned on a Spectral instrument. So I was kind of, I joined the lab and I was kind of thrown into these immune profiling projects, and I was given some panels that were designed for the ID 7000. So that was my entree to the flow world, and that was, the learning curve was super steep, but I've grown to really love Spectral for a bunch of reasons. So like in our case, we're trying to get as much data from a cell as possible in a limited patient sample, and so we're trying to understand like the full immune landscape in a person in response to infection or vaccination, and Spectral affords you the ability to do that. And I think we have some of the same challenges as Domenico, because we have CD4 panels and a B cell panel, and getting them all to behave is challenging. The other thing, like from after learning flow on Spectral, the ability to see the entire spectra of your cells is like, it really gives me a sense of confidence in my data that I don't get from classic flow when, large chunks of the spectra are obscured. So it's like, once you see that ribbon plot and you can see the full spectrum of your cells and all of your markers, it's like, it really changes things, and it's tough to go back. And third, like the ability to add more markers in Spectral, um, and now in that coupled with like these high dimensional analysis tools, like it allows you to like look at all these different markers, and and cluster based on those. So markers that were previously, seen as not important for some cell types can now be informative. And markers that we associated with one cell type are now like, unearthing new populations of other cell types.
So from our side at York, it's been really useful to hear that. But I'm going to have plants, so chloroplast, chlorophyll to show into the mix, and all sorts of different other autofluorescences off the 450, the UV side of things that aren't typically within the mammalian things. I'm going to have bacteria, yeast, algae. God, we're throwing all sorts at it. But even within your samples that you're mentioning, some of those cell lines have very different autofluorescences. And from my recollection, I think we can look at 10 different autofluorescence channels. So can I ask how many different autofluorescent channels you've tried, and has that been useful compared to one? Tamar.
Yeah, so far, just running whole blood, and red blood cell lyse, whole blood actually. I just tried to go for 4 different autofluorescent populations: neutrophils, monocytes, lymphocytes, and eosinophils. And especially eosinophils are like an old love versus / hate of me, because I've done my PhD. I've done a lot of research on them, a lot of flow, and they always look positive for everything because they're autofluorescent. And then when you go through these signatures, it turns out that actually most of these autofluorescent signatures are very similar. Like from the lymphocyte, monocytes, and neutrophils, they have the same signature, just a different level. So the brightness is different. But the eosinophils are really different. So they have a completely different spectrum. And if you don't account for that autofluorescence, you'll see signals. You'll think you're seeing signals that aren't there. And now with Spectral, you can actually extract that autofluorescence. So in this case, it's just whole blood, so it's not that complicated. But I already needed 2 autofluorescent signatures to get a decent unmixing. And I can imagine that if you run some plant material, maybe some water. I actually ran some water from the pond, and you see so many different signatures. And especially, for example, in plant biology, you might not have the reagents, you might not have antibodies targeting a certain type of cell that you're interested in, but you can even use the autofluorescence to just identify those cells. Because with eosinophils, I can now use the autofluorescence to identify the cells instead of having to use an antibody. So I think that's a huge plus for Spectral.
Yeah, no, and I'm itching to try some of those plant samples on it. I'll go Kelly, then I'll go Domenico. Kelly.
Yeah, we actually have a workshop for autofluorescence at Cyto, with Daniel Vosel and Rui Gardner. And I really want to like characterize a lot on the mosaic, because I think that the way that we're going to tackle this as a group, as a community, is by starting to qualify single sources of autofluorescence using single color autofluorescent controls. So for example, loading up a micelle with a particular autofluorescent compound, in order that it's no longer that we have different ratios of individual contributors. But if we can identify them as a single source of autofluorescence, then it doesn't matter if they go up and down, because I have an accurate understanding of the spectrum of that contributing NADH or FAD or chlorophyll or whatever that one thing is. Our issue, even with blood, is that once the minute that you go to activate it, you have 2 T cells that have a different autofluorescent profile, because one is now metabolically active and one is not. So it's a lot simpler when we're working just with healthy whole blood. You know what I mean? And even as Tamar is saying, that's still 2 autofluorescent profiles, let alone disease and chemotherapeutic treated, or all the different ways that we know that we're contributing to chemically to different autofluorescent signatures. So, I mean, I think that onus is again on us as users to start to create a library. What I want to do is create a spectral viewer for autofluorescence.
Right.
What have we done to that lung? How many, what is the metadata basically of that patient from which I'm deriving that lung sample? How many of those samples can we possibly have? Maybe some of them will all be the same. Maybe they won't. What are we going to learn? Will we be able to completely characterize, not necessarily, I don't think so much as label-free AI type learning, because I'm not positive that, I think there could be some elements of that become incorporated into our workflow eventually. But even right now, I can use it to say, this has a particular single source as a marker that I can relate to my antibody staining. And I think that is one of the biggest strengths of Spectral going forward.
Yeah, I think machine learning is the approach. Ultimately, what we're talking about is a machine learning approach towards it rather than just a black box for autofluorescence. And I think that we're going to learn more about our samples through that anyway. Domenico.
No, I'm just curious because I'm working with TILs from human lung. So if someone is just if someone is analyzing this kind of samples and notices that there is alteration in fluorescence, or how to manage the autofluorescence in this kind of samples, because I'm just starting with the Spectral now for this kind of samples. So, but generally I work with PBNC, so it's quite easy for us because we are interested in lymphocytes. And also, if you get inside monocytes, the autofluorescence so can impact, but not too much. But in analyzing TILs is quite different. And I'm just wonder if someone have some suggestion how to manage this kind of autofluorescence from these samples.
I think Kelly's solution would work in the long term, but go on, has anyone got a quick answer? An answer that's off the shelf?
I mean, right now, having those 10 channels of autofluorescence is really important. But they do need to create, I think that's one of the biggest strengths of the Mosaic, actually, is the fact that those 10 channels exist, because at least it gives you the backbone kind of instrumentation, the elements that are necessary, so that eventually when you have a software solution that can go in and compare those autofluorescent signatures, you're going to have this big wealth of all these different channels that you can do that in. But it does have to be an automated solution to characterize what is a unique source, what is a unique spectrum and what isn't.
Yeah, and you can actually, sorry. So you can actually use the when you unmix on the Mosaic, you'll actually get the normalized signatures, but you also get a similarity index for all your unstained parameters that you're defining. So if they're highly so usually how I approach it is just make as many autofluorescent signatures as you think you can find, unmix, look which ones are very similar. So if the similarity is like 0.99, you just remove one of the two. And you keep repeating that process and you remove everything that's really similar. And then in the end, you'll be left with a number of signatures that may be useful, and just try which one works best.
Okay.
I guess you want to do that with treated and untreated cells, diseased and undiseased cells. So it's quite nice you're talking about 10 autofluorescence parameters, and that's one of the advantages of it. Can I ask what your first impressions are of the Cytoflex Mosaic? I'm going to go to John first. Sorry, Andrew, either. Go on.
So my first impression of the Mosaic was I really liked how the software is identical to the Cytoflex S and LX. I started on a Spectral instrument that was much more complicated and more buttons and sort of features, I guess, to learn. And so I really liked how it really wasn't that much different from running the LX. I mean, the software is identical. I think that's helpful for researchers who start their pilots on the S or the LX, and then enables them to upgrade to an instrument that they're familiar with instead of having to learn this crazy new spectral software. That's what that was my first impression. I was like, oh, this is like the LX.
How easy is the software? It's very simple. It's very easy. It's incredible compared to the other ones that I use. So it's very, very easy, fast, and also for the youngest researcher in our lab is very. They are are able to use the instrument after 1 or 2 hours of just training. So it's very, very fast, similar to the conventional one. Incredible. So I was very, very impressed regarding this kind of work. So, and also is stable, and the implementation of the 2 algorithm was great. The transformation is amazing. So one of the best I ever seen. That's my opinion.
Good strap lines there off your Domenico. So we'll get you a T-shirt that says it. Amazing, yeah. Incredible. Andrew, you were going to come in with something.
Yeah, absolutely. I mean, as for anybody who has worked in a core facility, the biggest issue that we have is trying to get people to use new instruments and just having that familiarity of site expert right away. They can move on to the other instrument without having to worry about, obviously we're learning a new concept here in Spectral. Removing the worry of learning a new software is going to be incredibly helpful when it comes to getting new users onto the instrument.
Okay. Tamar, you see any other? So I've got one thing that I'm really, really pleased about. But go on, Tamar. What?
Well, so I've never actually worked with a Cytoflex, and with we have Cytoflex SRTs, and the only thing I was able to do, I've done so far, is run a QC on it and start it up for the users. But within 2 hours, I was able to run an experiment. So I think it's pretty easy, to run. Yeah. So some of the unique things that I like the infrared laser. I haven't, because well, we were talking about autofluorescence before, but if you want, preferably you'll do your measurements. If you have a highly autofluorescent population, you'll do your measurements in a place of the spectrum where there is no or very little autofluorescence. And I think that's in the infrared. So now that you have an infrared laser, you can put some important lineage marker, so that you can identify your really difficult cell of interest with a marker and with a fluorochrome that's not in the autofluorescent area. So I really like that bit. And it's also a few extra colors, so it makes the eyes happier.
So we've got a couple of LXs and an S, and I've got to say, and we've got other cytometers as well. I think my biggest concern about Spectral full stop is buying a new system so I have to look after my operating costs, and a new system comes with quite a big operating cost. So that's quite a big weight around my lab budget, and not everyone needs the Spectral. Yes, I think once we have a system, even if it's 2-3 color, they'll probably end up just doing Spectral, but not everyone needs it. So actually writing a grant justification to buy it is really, well, actually I've said no to it. But of course, this is a Mosaic Spectral module that actually just sits on the side of our existing LX, or you can even hotbed it between 2 LXs if you want to, if you just got the one module. So the price it's now in the realm that I can afford, well, potentially get funding for easier than having to write a big, very competitive application. But the operating costs should be less. Contrary, if you're listening, keep them low. Because that's a really important aspect to it. But it does mean I've now got a Spectral, and I haven't got to have another Spectral and all the other contracts. So for me, actually, this is really the financial side is really attractive. I don't know if anyone's ever thought about, I don't know how all your labs are operated, whether that's a key aspect to you or not. Anyone like to
Can
comment?
I add to that?
Yeah,
Because
Kelly.
I completely agree with you. I think that, we don't really know what the next 4 years look like. And the fact is, I think it's almost genius that they decided to have a module where I don't have to buy a whole new instrument. Like anybody that knows me knows that I don't blow smoke. Like if I didn't think it was good, I would say so. The thing that I like about it, and probably the more unique perspective that I have from being from the manufacturing side of the business, is I was, maybe I'm just getting old and a little bitter or something, but I just get kind of tired of companies releasing instruments that then I'm supposed to optimize and I'm supposed to handle. Like I remember when the Aurora was released and the software didn't work for those of us that had the first instruments, and we thought we were going crazy and nobody told us. So then you're just wasting reagent and time and your patience, with something that's not working. And one of the things I love about Coulter is this kind of clinical mentality they have. They're not going to release something unless it's ready to go. And I think all of us are seeing that, we got our instruments placed. I made panels to the best of my ability, not understanding how, the instrument was going to perform. And we're not going to have to go through 5 iterations of these panels. It's going to go into a core, and you can already tell you're going to be able to transfer panels with very little effort between an Aurora and the Mosaic. And you're going to do it with an adapted, like a component to an instrument you already have. Like, I'm just really excited for them. I feel like they made a lot of good decisions, and they did the work beforehand to make sure that when it's released, I'm not, responsible for,
Yeah.
the rest. It's like sometimes as a user, you feel like you're finishing their R&D process. And that should have happened before I bought it. And that's not going to happen with this instrument, well, to the best of my knowledge.
I've been there plenty of times, including with Coulter in the past. But actually, I think a lot of these companies are now much better at getting things to market right. But do you know what? As a beta tester, it's what you expect, isn't it? It's what I like, is finding the problems. Tamar.
To be honest, I think like most of the users of our core are already sold to Spectral. So for my core, I'm not sure whether the Mosaic, like the add-on, is perfect, is what I would get, because we have our Spectral instruments are already run until midnight almost every day. So I'm not 100% sure whether for me the Mosaic would be the best choice. But if you're a smaller lab, you don't have as many instruments, then I agree it would be a very good option. Yes.
You can imagine, Tamar, if you had 3 Cytoflexes already in the lab, and you want
it's a great upgrade.
Get all your systems on Spectral for less than the price of one.
Yeah, well, I'm already. Well, if I can get a Mosaic for my SRTs, that would be fantastic.
Again, Coulter, are you listening?
I'm sure I'm not the first one who's asked.
I know, you just can't be happy with what you have. You're like, what's the next thing? Give me more.
More. Yes, of course.
But Tamar, you are really sure that you should need to upgrade your SRT to Spectral just to sort cells?
Yes, absolutely.
Yeah.
So there's already so many users that, they're all running their 30, 40 color panels, and they're already saying, well, so for one thing, they have a funny population, and they're just asking, like, I'm not sure what this is. And I was like, just sort it. Each time you're not sure what a cell is, sort it. Maybe make a cytospin of it. Just look at it under the microscope. But also for index sorting, if you're doing that type of a study, just single cell, in each well you have a cell, and you can do 30 color index sorting. Well, maybe I'm not the one who does it, but there's always going to be that one person who needs it. Absolutely.
Yeah, no. As soon as you've got a Spectral panel, you want to sort on it. That's yeah.
So I agree 100% for the index sorting, but I really don't like to push much more than 10, 11, 12 core for just sorting. So I think the less markers you have, the better is your sorting strategy. But anyway.
I want to add to that, because I think that the deal is not necessarily that it's that you don't have to build a separate panel for your sort, right? Like you could take your 30, 40 color panel and you could reduce it to just the markers you need to sort. without having to completely redesign the panel because you're using fluorophores that a traditional instrument wouldn't see. So I agree with both of you, because when I sort, I want to be sure that I'm really kind of getting the cleanest population possible, and maybe that's not going to be at 30 colors. But I also, as I've been developing some of these panels and we're trying to compare them to the traditional Cytoflex, you have to build it for the Cytoflex first, and then you try to adapt it to the mosaic. That's not the best way of building a panel. The best way to build a panel is to build it for the mosaic and then reduce it, based on performance measures, so I agree with both of you.
Yeah, and ultimately, if you can sort and analyze at the same time, you're getting double value, because you're not just sorting, you're also getting data to analyze in a different way, potentially. But we're not getting a sorter at the moment. That's not here yet. So don't get excited or too excited about it. John, Andrew, how many different users have tried it at the Broad?
As of right now, it is exclusive to us. We do have people using the instrument occasionally if we ever need a backup for Cytoflex, if they're all booked for the day. So I have had a few trusted users give it a crack, but as of right now, it's just been us testing the actual spectral component of the instrument.
So, okay. So does that mean they can't use that system at the moment?
It's just not bookable. So we're just keeping it to ourselves so we can run our experiment in our panel.
So we've even got our 3 Cytoflexes. We're still switching it. So actually, as soon as the engineer had left, we switched it back into conventional mode. And just using the 21 color detectors off it. So the next user who we haven't trained yet to use it could come in, use it as they're comfortable with. We're doing a course coming up next week. It starts tomorrow. Running a course on the basics, the fundamentals, and moving into advanced flow cytometry. So the first 3 days is in conventional mode. But then I can switch it Friday morning, just switch it over, and then we can do a spectral. But we won't this time round. But that's the potential that actually that's super good. Because it means I don't have to retrain 100 users. They can just, when they're ready, they can softly migrate over, start using it, and then tell their mates that it's better, and then it will organically grow, which saves a lot of staff resource time. But Tamar, how many users are you getting on yours?
So far, it's been mostly for the staff, indeed for testing. But I do have, I think, about 4 users that are trained on it by now. And they're just, they're flow nerds like me. So they're the ones that are, there's a new instrument. Can I try it? And then like, I'm running a panel anyway on the Aurora. Let's try it on the Mosaic. And they're migrating pretty quickly. So, and I think they're pretty easy to train so far. So they know of all the potential problems with Spectral and all the difficulties. They've been happily using it.
Domenico, similar things, or you've got other users using it?
So we are just 5, but I'm the only one that are using now the Spectral. I'm the only one now in our lab. I will train our students so far, but now I'm pretty alone.
Yeah, so I think
it's a paradise, not just for the moment, but the problem is that the rest of the staff is just using the conventional one, so I have to switch manually every time. It's a little bit tricky. I'm not so happy just moving the fiber from one side to the other one. But anyway, I will just take my time, do the switch, and then I will run all my stuff.
Oh, I've got a good question. How many colors have you actually pushed up to? What is the max you've tried on it to date?
Now 21.
21. Okay, yeah. Tamar, how many?
Myself, 30 colors. There's one user who's done 38.
38. I'm presuming, Andrew, John, are you working with Kelly on a big panel?
We are. I forget how many colors exactly, but we ran it a couple weeks ago, and it looked mostly good, right? We just had to make some adjustments, and we're about to give it another go with the changes.
So we'll have 29 colors on Thursday, and that will be pushed to 39 colors in a probably couple of weeks here.
Tamar, you'll be one behind.
Yeah. I just had to look. I had to look at the panel and count backwards. But I think we have 36 for an ILC T cell, like non-conventional and conventional T cell panel. There's definitely some areas, like we've been struggling a little bit with per-CP, but I don't really want people to use per-CP anyway. So I feel like it's okay if that one doesn't work.
Well, Kelly,
That's
what are
not
you
my
going to
most
be
precious channel. So
Kelly, what's your alternative to per-CP?
oh yeah, I mean, so far all of the work that they've been doing on the real blues and the real yellows, they're just really wonderful dyes. They do, I think one of the vials was 12 micrograms of antibody. So I think that's a question that we should all be thinking about. Is not a lot of antibody in that vial. And the background was a little high because brighter things, even the background is brighter. So that's the balance of really bright dyes is, do you need to be at saturation? Because you're not going to be at saturation with those. Maybe, that's actually my only hesitation so far is just that we've had to titrate down so far in order to combat that background. And if you don't, then now you don't have, if you're not running all your good controls, it's hard to track that background in your fully stained sample along with your increase in spreading error. So, that's been one of the ones for me that has been questionable. But still, we're, the Brilliant Violets are going off patent soon. We're just going to have an explosion of, the polymer dyes being accessible to any generic manufacturer. And it's going to be a lot. It's going to be a lot to remember, like for the individual user to have a relationship with dyes and all these different, marketing names, all the family names people are going to assign to the same dyes. I think that's going to be a challenge we're going to have to face going forward is what's equivalent, what's better, how do we qualify that?
So the software comes with two different algorithms, I think, and that bit I haven't played with at all. So does that help in those scenarios? Have you tried both algorithms? What are the pros and cons? I have no idea. Please help someone.
So I've tried both. Well, every time I unmix, I try both algorithms, to be honest. And I know from publications that the Poisson unmixing is supposed to give a lot less spread. But to be honest, in my hands, I've had a few cases where it gave more spread. So I'm not sure if that's just because of how I'm using it or whether it's a software implementation. But I have also seen that sometimes the Poisson unmixing just gave me better results, so less errors. So I think it's just, you'll have to try it. Like so it just gives you another option. So in my cases, it wasn't actually better, but I do like to be able to try different things and just see what works best for my samples.
Okay.
I think it remains to be seen to characterize what condition it needs to be applied to. Like we've all only had access to the instrument for a few months. So we really haven't been able to put it through and characterize, each of the elements of it that are going to need to be optimized. It makes sense that it's going to have better by only the math, but I don't think it's going to be a generalized statement. And I think culture needs to be careful about making any generalized statements about things. We know how complex flow is. It's better to show when and where and under what condition it's helpful. But I do like that they have it and that because we all know that, unmixing and, spreading error, it doesn't have to be that way. Theoretically, that is absolutely an area of growth that people need to be paying attention to. And I don't think anybody on this call is going to be making new algorithms. So somebody needs to listen to us.
Can I just say, I take it it's just as easy as you get your data, you try algorithm 1, and then you just click algorithm 2. Is that right?
Yes.
Yeah, because I'm listening thinking, this is sounding daunting. I've got 10
I don't think you can live unmix Poisson though, right?
No, post, surely.
Post, I think, is the only, because I think it would take a little too long.
Yeah,
I'm not positive.
Yeah. I don't think I'd want to do it live because you commit. So you kind of want your data and then just click and process the data.
But that's the beauty of Spectral. You can, like with these instruments, you can even I forgot all my single stains, but you just run the panel do your single stains tomorrow, maybe, and then you can still unmix. So I wouldn't recommend it, but it may have happened to a friend, you know. But you can do that, and I think with the poisson as well. So yeah, I haven't actually tried to run it live, but yeah, afterwards you definitely want to try these things.
Well, but that's really key point. The fact you can get another control the next day, if it fails on the day itself. And there's instances where users cannot get easily single label controls for whatever reason, especially when you start going into plants and other materials, they just cannot get that material. So actually we have in microscopy, we go and find another user that has got that color in their sample. And we use that. If it's a very strong signal, the autofluorescence is then insignificant on the back of that very big positive. And we can use that, which then enables them to unmix that they wouldn't otherwise have been able to unmix on. So I presume the same can be done here.
So why don't you just use beads? Why bother finding another sample?
Can you get GFP, CFP, YFP, M cherry, Cerealia?
Okay, all right.
You can actually just use the spectral library. So you can add like all the single stains that you ran, you can put them in the library and you can reuse them for the next experiment. I'm not sure how that will work if you reuse a control from a year ago. I don't think that's a good idea, but you can reuse them.
Yeah, in conventional works good.
Just that uniformity across the detectors and sensitivity across detectors, that is going to be a variable over time that can happen. But for dominant signals and the way the algorithms are weighted, some of these can...
Very simple panel. Yeah.
Domenico, you're going to say something? Sorry.
Yeah, just for conventional one works good, but for spectral, I don't know. So if you can use the oldest single stain control, I would like to test because it's very easy to import, export all the stuff in the Cytoflex software, and could be great test this stuff. Also because the instrument use the ADP that make the signal linear, and probably, I don't know, I'm not sure 100%, but could work. But in conventional 100%, so I tested it. More than one time, I forgot to prepare one, or I had a problem with one single stain control, and I just click on with the right button and import single stain control from other work experiment, and it is still work.
I mean, if it was as simple as using stuff from a library, we wouldn't use single color controls anymore. And I can only imagine how not good that would be. So I definitely think that we shouldn't be encouraging people to not be running single color controls. And because they had been kind of promoting that on the Cytoflex previously, it worries me that they'll continue with the same mentality of trying to promote that behavior. But I think that there's two things here. Simple organic dyes and extremely well-made tandems are much more stable than they used to be. But if you're going to use PE-Sci5 and ECD and PE-Sci 5.5, if you're going to use those, you're not going to have the same signal. It's just not, that's not how it's going to work. But if you're using, I made the PE Dazzle 594, the degree of labeling of that acceptor to donor is way different. And so it is more spectrally stable to small variations over time. But do we rely on that? Because a bunch of us are doing the work to show the photo instability of tandems. And that's just photo instability, let alone all the different fixatives and all the things that we know are degrading our tandems. So the thought that you could use a libraried floor spectra on a fixed sample, those two things absolutely do not look the same. I've done so much work to show tandem degradation in that instance. So I don't want us to ever make general statements about libraried floors. It gives me goosebumps.
Yeah,
because I sit in some of these like meetings where I just see people putting data on the screen, and I can't understand why everybody isn't raising their hand to question what they're seeing, because they see populations that are just sprouting like little buds off of every part of their populations, and nobody is holding them to a higher standard. And stuff like that will manifest when you're using library floors.
No, so
I feel like the old lady in the room here.
absolutely
I'm like,
agree that you should match like sample type and everything. But at least there's something there. And indeed, like if you forgot to add the antibody to the single stain or something, at least you can run the control a day later and have an ID. I totally agree that you should. The best thing is to use fresh controls.
And yeah, tandem dyes were different beasts compared to your Alexa 488 or your fluoresceins or your non-tandem dyes, which are less gregarious in their, is that the right word? No, less variable in their emissions. But again, we have to be careful with pH changes in our samples. Good grief, how many people use DAPI? And as your DAPI stock ages, the fluorescence of it can change quite dramatically. You know, I've seen DAPI off a 488. Yeah, and
that's actually called homophredding. It's not necessarily the degradation of the DAPI. It's that in the DNA, the fluors are so close they start to fret together and they shift green.
It's an age issue when you get fixed samples as he gets older and older.
Oh, yeah, totally.
The microscopy, see it, it's like, oh, and then I'll say a particular vendor. And go,
you're talking about DAPI in the mounting media, right? Because it's pulling all that DAPI into the DNA over time. Yeah,
but I think that's what's quite nice, because you can see the spectra. You can also see these changes in your data. You can see if something is wrong. Whereas actually, if you're just doing straight compensation, you wouldn't see these changes. So it's these little benefits that we're getting. And then the more you engage with your fluorochrome, seeing your fluorochrome, actually having that on the Cytoflex mosaic is going to be super useful, I think.
Something I think that we'll have to make an effort to do is to like really kind of educate users. Like if we're going to make Spectral easy, if we want to make Spectral easy, we have to, people need to know how to look for errors in their unmixing. Like, you know, Spectral can be exhausting. They're long days of staining. It's long periods of optimization. And by the time you run your samples, you just want to get your data and get out of there. And I was taught, like, you shouldn't even begin to analyze your data until you've assessed the unmixing. I don't think 90% of people who come through the core know how to analyze their unmixing, including me. Like, how do I know that little bleb or that bud that Kelly was talking about is not some error or some, you know, anomaly or something or artifact and not, you know, some cell population? How do I assess that? And I think that we have to consider that, like, when we're designing the software. So just
how do we ensure the data, the quality of the data? How do we do that on the side? How is the Cytoflex mosaic enabling us to check the data quality? I see Kelly, you just unmuted. Go on, Kelly, and then Tamar.
Well, they do give you the N by N is the most important thing that you're going to look at, but you need to be able to assess the N by N by each mark, by each control. So they display, I'm pretty sure, I haven't quite done this yet, have you guys? But it should auto-populate an N by N, and then you flip through your samples and it displays it across each of the detectors. And that's the first thing, before you even leave the instrument, do that, because, you know, I agree with you, Peter. When I do Spectral, I don't do live unmixing because I do, you know, we need this data. So I've got the, you know, 2 different single color controls, my biologic and my bead, you know, and I still need to choose which of those is the best. Maybe my bead got contaminated. It's super easy to contaminate beads. You don't need a lot of contaminating antibody. Those beads have a high binding capacity, right? So just relying on them isn't necessarily the best way to go either. So yeah, I think it's really important to kind of get your data displayed first. If you only have your fully stained sample, it's too complicated. There's all these things going on all these different cell types that is not helping me identify where my problem child is. And it's a lot faster if I do it one by one. And I think it does have like an error message. You guys might know this better than me. My instrument in Loveland here in Colorado at the Coulter site, it hasn't been accessible to me until like this week. So I don't have quite as much hands-on time as these guys do, but I think it gives you an error if it thinks something's contaminated, right?
Tamar?
Yeah, I think there's an error message, like there's a deviation from the mean. which just means that if your single stain, like if you don't have if there's some events in there that are different from the mean, it will give you an error message. Also, if there's oversaturation for some of your events. Yes.
Anyone else got any thoughts on this?
Well, there's one more thing that I quite like. So the N by N plots are definitely something you should do to assess your data quality. But I really like that when you unmix, you get an image of all your normalized spectra. So you can at least look at, like, I put in an APC. Does it look like an APC? It will also give you similarity indices. So if you have a little bit of experience with Spectral, you get a feeling for what these indices should be. And if they're completely different, if the normalized spectra are completely different, you know something went really wrong. And that's a really helpful feature.
The other thing that I like about that, Tamar, is the spectra actually looks like spectra. It annoyed me so much. Like on the ID7000, I would look at, I study fluorophores, and I would look at those spectra and be like, I don't know what that is. You know, like how they displayed it with the wide margin and the heat mappy kind of thing. That's not helpful. So I love it when they do. I think you called it a ribbon plot, John. You know, like when they, have something that I'm more accustomed to seeing, I'm like, oh yeah, it's an easier, visual for me to say, I'm pretty sure I know what that is.
We have 5 minutes to go. So I'm going to come to Andrew and John. How many spectral systems, other manufacturer spectral systems do you have?
Three others, if we want to count Bigfoot.
Okay.
Sorta. So yes, 3 other instruments.
Would you switch them all out for a Cytoflex mosaic? Except the Bigfoot. So an SRT mosaic when that comes out.
So the S8, probably not, considering it's a sorter and.
But you've got the imaging side of that, yeah.
As well. If I were to choose, probably yes, considering my users' familiarity with the CytExpert software. I think that would be the best fit for our core, or we could just add 6 of them to our Cytoflexes. But yeah, I would say that I prefer them to the others.
It was a very difficult question. Sorry, John, are you in agreement with that?
I think once people find out how easy it is to move over to the mosaic, that thing is gonna be booked constantly, because it's just pretty effortless to make the transition. Just remember to hit the power button on the module. That's something that we forget to do all the time.
Thank you for that tip. That's a really useful tip for us. Domenico.
So we have also the Bigfoot.
You don't have to name and shame or do anything else, don't worry. But
sorry?
Are you so happy with the Cytoflex mosaic? You would be happy if that was all your spectral options.
I'm very I'm very happy with this stuff. So so I will just say thank you to the Beckman team for the collaboration, because it's for us is a great improvement. So it's very easy to set up for us and compare to the other ones. So I really love with this kind of instrument. So I just need now so I'm just waiting now for the new set of antibodies just to go ahead with our experiment. I'm very happy about this collaboration right now.
Domenico, thank you. Tamar?
So I do like to have a bit of variety in my core. So sometimes one instrument might be better for one application than the other. So we have 8 spectral instruments right now. So I'm not sure if I would trade anything in, but I do see the mosaic becoming like a workhorse for us that's fully booked every day.
See, that's like Christmas when you get a box of sort of chocolates and it's an assortment box, but there's always going to be a favorite that goes first. That's what's going to be booked out on your booking system, isn't it? So it'll be interesting to see how that goes. And actually, yeah, obviously we can't really talk about the S8 on this. We're talking about the Mosaic, but another actually really good product. The imaging side, yeah. Do you know what? We're really lucky in the flow world at the moment. You know, all the companies are really chasing each other, and they're always going one up. And I think we're really fortunate right now with the Mosaic that I think it's going to make a huge difference to us. Not having to invest in whole analyzers is just mind-blowing. The fact that I can now afford to get these on and multiples of them, should we want them, is just... And with the finances in universities at the moment, you know, this could not be better timed. I don't know. Culture just seems to have hit a sweet spot on this.
And generally for the environment. I get so tired sunsetting my instruments when they're still perfectly good, but they just they're not going to have a service contract anymore. Like, I love the idea that you're taking instruments you could have had for 10 years and you're just making it better. Like, that's what's good for the environment.
Which is true, actually, in the UK, actually asking to get a replacement system with new capability, not a problem, but none of my systems are out of date. So actually, to say I need another system, all I'm doing is diluting my user base, which is a big thing. I'm going to ask some, because we are up to the hour, gosh, really quickly, some really, really quick fire answers. So yes or no's, not quite yes or no's. Difficult or easy? John?
Easy.
Andrew?
Easy.
Domenico?
Super easy.
Super easy. Okay, cool. Tamar?
Easy.
Kelly?
Yeah, easy.
Yeah, I thought you might say that. Compensation or spectral? Kelly?
Oh, Spectral.
Tamar?
Spectral.
Domenico?
100% Spectral.
John?
Definitely Spectral.
And Andrew?
I don't want to be different.
Okay. And the biggest benefit? This time to Andrew first.
Ease of use and adaptability.
Cool. John?
More information from cells.
Nice answer. Domenico?
A lot of markers to analyze, a lot of information, great quality of data.
Oh, how you come up with different answers for each one? Tamar? No stress.
Just so many markers. So I love it.
Who have I missed?
Me! I would say
Kelly.
transferring panels. I think that's going to be a big thing. We've always complained about transferring panels, and now we'll be able to from the Aurora to the Mosaic.
What awkward question. What's on your wish list then? If you could do one other thing, Andrew.
A switch that makes it switch between spectral and conventional.
Yeah, a little easier than just take it. But I've got to say, the whole thing took us minutes.
Yeah. It's just my poor fingers having to plug in the cables every time. It's a little tough for me, but it's simple enough. Just a small nitpick.
Yeah, and do you know what I think the brutal reality is? As much as I love the fact it's an add-on, I think once it's in spectral mode, it's going to be rare it goes back. Especially as the users, once the users are all onboarded, it'll become less and less, I hope. John?
Like
Biggest
practically
benefit.
or dream feature. Let's see. Yeah, I guess probably the easy switching, less manual kind of plugging things in and out, I guess. But
So
also, like, in the future, I would like to see some sort of like AI correction for spread error. Like, why can't we predict that and remove it?
I like that. Domenico?
I agree with John and Andrew. So make
Okay.
it easier to switch between conventional and spectral.
Tamar, I think you've already answered this.
No, I have a long wish list. I always have, but I think autofluorescence finder. Just I put in my unstain, and it will tell me these are the autofluorescence signatures you might need to take into account. I would love that, and a bubble detector, so I can run it up in plate mode, and I get that last cell out.
I thought you were going to say a sorting mosaic.
Second one, third one on the list. Yeah,
Kelly?
Yeah, I'm with Tamar. It's all about the autofluorescence for me. a lot of the unmixing and a lot of the spreading error that we're seeing is in areas of high autofluorescence. And I think that all those things are tied to one another. And I think they need to have it by the end of the year. Like they maybe don't need it at release, but it's that important that you need to have a solution soon.
Do you know, you all made me think. We just put in a grant application in at the moment, and we're going a bit off piste. But actually, having the Spectral on there for actually think all that marine life. I can't remember who said they got plant water. Was it Andrew? You said got water, or Tamar? Who went and got water from the pond?
Tamar.
Tamar, you said you got water from the pond. Just to be able to look at the autofluorescences and then look at the stress on those cells, because those autofluorescence parameters will change. We will be able to study it just with that. So again, my head, yes, we've got a lot of immunologists, stem cell biologists, but actually it's all that other work that we can do with it that is super cool. Tamar.
Well, you also have 6 side scatters. I have no idea what to do with it yet, but I'm really curious what everyone's going to come up with. It works for eosinophils as well, actually. So, but 6 scatters.
Yeah.
Oh.
Let's see what we can do with that.
Yeah, but just imagine you could. Oh no, I've got yes. But don't forget. No, thank you. I've got some ideas on that. I'm not going to mention it here. Sorry, guys. I'm going to publish that one first. Everyone, thank you very much for joining me today. Thank you, everyone who's listened to Flow Stars' special. You've heard of some of the previous guests. Just hear about the guests themselves, like Rui Gardner that Kelly was mentioning earlier. Please go and tune into those. But everyone, thank you very much. I can't wait to see what happens when we get to the next Cyto and seeing the results that are coming off it. I can't wait to be showing you our results of all the weird and wonderful stuff. Not just blood, but other stuff as well. Anyway, thanks, everyone.
Thank you.
Thank you.
Thank you,
Bye.
Peter. Thank you, everyone.